thank you for that. MIT is actually where I did my PhD training and postdoc training. This is a picture of me there. I was a student with Graham Walker studying basic mechanisms of mutagenesis, actually discovering the function of a gene that Cynthia Kenyon found long ago. And we would do experiments that would have data like these, where we would see green dots, and we would say that that was a site of functional localization within the cytoplasm in response to DNA damage. And then for my postdoc, I crossed the street to the Whitehead and joined Susan Lindquist's lab. It's an amazing environment. Simon Alberti, who you'll hear from in a moment, was my, not baymate, but in the bay next door. And we would see data like these, and we would interpret them as being aggregates. And it seemed weird to me that the fundamentals were exactly the same. And kind of at the same time, it became clear that the picture was far more complex. So Simon wound up going to Dresden and thinking a lot about liquid properties of condensates. I began to meet Ibrahim, who then had much less hair, and see his data and think about all of this. That's true. Fair enough. Yeah, fair enough. Yeah. And then, actually, Phil hosted Steve McKnight to give a very provocative talk in the Whitehead Auditorium, arguing for the role of gels that might be formed by many intrinsically disordered proteins. Okay, so in my lab, and in fact, starting when I was in Susan's lab, I was very excited by the idea that protein folding might be far more complex than we're taught in textbooks, where the polypeptide sequence of one protein gives rise to a particular fold, another protein to a different one, but rather that one might give rise to many, and that they could in fact encode information. Indeed, we now are well aware that a lot of proteins in us and many organisms look very different than what we're taught in undergrad biology. They're appended with large disordered domains that we like to invoke could do anything that we want. But one thing that I will hopefully convince you of is that they can assemble or allow proteins to assemble into multiple conformers, at least some of which can actually template their own fold onto newly synthesized proteins, and in so doing, create kind of a way of transmitting information across generations in just the same way that the replication of the genome can. So this story begins centuries ago with this sheep that is sick with a disease known as scrapie. You can see the phenotype from actually scraping here. It baffled molecular biologists, veterinarians, even people in the House of Commons centuries ago, right? Because the patterns of transmission were just so bizarre. And of course, we now know that's because it isn't driven by a virus or normal pathogen, but rather from an endogenous protein within the animal that's undergone a change in its shape and one that is infectious. Okay? And that is present, that basic behavior is present actually throughout life. So much of what we know about it comes from these two plates of yeast found by Tuitt and Cox in the 70s, really paying close attention to detail. And something is obviously different about them, even though they have exactly the same genome in every way that we can discern. And what's different is that they have a different proteome. Okay? But not in a way that you could tell very easily if you did a mass spec experiment. Instead, one protein, a translation termination factor, is in an aggregated state, it forms its assemblies, in the white cells. And you'll hear more about actually the very complex life of this protein probably from Simon in a moment. But these dots turn out to have unusual behaviors that allow them to transmit information. If you take the protein and purify it, it can actually assemble into a variety of structures. But here, with agitation, it forms these beautiful cross-beta sheet amyloid fibers. And they have the power to seed. So you follow with an intercalating dye like thioflavin T, you see a long lag phase followed by an assembly phase. But if you take a little bit of that material from the end of the reaction and use it to seed a new round of assembly, you see something totally different. Instead, a burst. And that comes from the faithful templating of the aggregated form onto oncoming monomers. So that gives you kind of the first part of that cycle of replication. It's more complicated in cells, right? So those are kept by a variety of spatial quality control mechanisms in the mother. But it turns out that the activity of chaperones can fragment these fibers into smaller pieces that we call seeds. And these are the real replicons. They're passed to daughter cells where they launch new rounds of assembly there. Okay? And this is really it, the cycle of fragmentation, rebuilding, of templated addition that allows the information to last longer than the half-lives of the individual molecules that encode it, in the same way that we see with the genome. Okay, and because of this, they wind up having very strange behaviors in genetics. They're dominant in diploids, and they defy Mendel's laws because they're not attached to chromatin, so they're passed to all meiotic progeny. So this is an exciting topic. It's motivated 2 Nobel Prizes, but seen as paradigm shifting, but frankly, very rare, and always driven by that amyloid conformation that I showed you. But of course, contemporaneously with me starting my lab, it became clear that nature is an amazing materials scientist, right, and can do extraordinary things by putting protein and RNA and DNA together in ways that we might not have naively assumed were possible. And so we wondered if there might be other ways to have protein transmit information. And what we capitalized on was the realization that actually in all of the biophysical studies of protein self-assembly, the assembled state was more thermodynamically stable than the native fold. But there's a huge kinetic energy barrier to achieving it. However, if you make more protein from the get-go, you can increase the likelihood that within a given cell, you're gonna adopt the templating fold. And because it's kind of its own catalyst, once you get there, all of the protein is converted into the assembled state. And then because of this heritability property that I just told you about, within that lineage, you're gonna keep maintaining the altered conformation. So we took advantage of the fact that in yeast you can do anything that you want. And we took every open reading frame and overexpressed them individually and transiently. And then we asked whether that led to an enduring change in the behavior of the lineage derived from this. And then we did a bunch of tests to show that basically it wasn't from mutations, that it had these kind of funny inheritance properties that I just alluded to. They defy Mendel's laws, and if you change the activity of chaperones even transiently, you can erase these traits in a way that you would never get for a mutation. Okay, and in so doing, we found dozens of new prions. I'll tell you about them. They're typically beneficial. They're driven by disordered proteins. They tend to have a conserved templating property from microbes to humans, where we've looked at it. And most of them don't form amyloid. So this is a semi-denaturing agarose gel that allows us to visualize the fibers made by the Psi prion I just showed you. And we don't see really any of this. Instead, they form condensates of various kinds. You'll see some examples. And I think the other big surprise to us, given that we think of as prions as subverting the central dogma, was that almost all of these factors that we found feed back onto earlier stages of information decoding. Okay? They turn out to be regulated. When I started doing this, I thought of these as errors in protein folding. But almost every one that we've looked at in some detail, they arise spontaneously at a frequency of 1 in 100,000, but they can be induced by very defined signals, whether that be interactions with competitors in the environment, prolonged cell cycle arrest, replication stress, or even interactions with specific nucleic acid targets, as we've heard a little bit about here. Okay. So I tell you first just a story about a role in transcriptional regulation, really epigenetic control, that was led by Zach Harvey when he was a student in the lab. He was captivated by one of the strongest hits of this class. That's the scaffold for a histone deacetylase complex that ordinarily keeps yeast from engaging in meiosis. This is kind of the most complicated thing that they're ever going to do. And the complex exists in various forms in all eukaryotes, and the idea is basically the same. You repress a developmental program that shouldn't be on all the time. And what we found was that from this transient overexpression experiment, we got a change in the conformation of the protein. So it became more stable. Here you can see in this partial proteolysis, just visualizing a MIC-tagged version of the protein. And that stability could be templated. So if we would take just a small amount of the lysate from the cells that have the stable form and dope it into the naive form, we could seed the stability and keep on going. It's like a PCR with protein conformation, if you will. And that was coupled with a change in phenotype. So even in bulk histone preps, you could see an increase in acetylated H4 and H3. And as you might expect, also that they were less able to repress the meiotic program that the wild type could. When you purify the protein, you start to see funny things happen immediately. So basically, after a couple of hours, even just coming off of a nickel column, the protein would form homogeneous gels in the column, akin to what McKnight wound up seeing with his isoxazole. And those gels have this capacity to transmit information. So you can see them in vitro, but you can also sonicate them and expose naive cells to them. It's a little bit of a trick here. We use a carrier plasmid and capitalize on the fact that a cell that's competent is likely to take up more than one thing from its environment. Basically, when we score for those that were transformants with a carrier plasmid, we find that about a third of them also have acquired the prion state. Okay? So again, we don't know whether that gel is the real thing, but back when I was a mechanistic enzymologist, we used to ask if it was kinetically and chemically competent to proceed to the next step, and this tells us that that material is biologically competent to drive the inheritance of this state. So like most of the proteins we found in the screen, extremely disordered and replete with phosphorylation sites, we were intrigued by two at the C-terminus that were found by David Morgan and Liam Holt to be phosphorylated only after a prolonged G2M arrest. And we confirmed this was the case. And we just asked if that would be enough to induce this element. So again, the spontaneous switching rate is about 1 in 10 to the 5th. But after this conversion, basically 85% of the cells have the phenotype and also have the associated change in the SNT1 stability. And as a frame of reference, this is about twice the frequency of kind of Waddington's classic ether experiment, so it's a strong effect. So what's it doing? Read-write mechanisms give you a capacity to maintain repressed chromatin over quite a number of generations, basically putting the old mark onto new octamers that have been deposited after replication. But open chromatin has been more mysterious, and some people, like Danny Reinberg, have even argued that basically we shouldn't consider it epigenetic because there isn't such a mechanism. And I think it's sort of provocative. I also thought that was true. But this is another way in which you could do this without a canonical read-write. But how does it happen, right? So we call it easy for an interesting reason. So like many such complexes, there are sort of conflicting roles. So one of them is repressive, so taking off this acetylation mark that I've told you about. But also it can recruit RNA polymerase II, that's an activating function. And indeed what we found in a variety of RNA sequencing experiments was a shift toward more production of RNA. However, they were all expressed at a low level. We didn't know what to make of it. We thought it might be an artifact. But it turns out that they're all derived from subtelomeric regions in what are called Has domains, basically about 40 to 50 kb from the telomere. And it's an emergent property that you don't see actually in any of the individual deletions of components of this deacetylase complex. We could show with reporters that it was real. And so we started doing CHIP, and we found that indeed both of these behaviors are localized there. So you see more acetylated H4, and you also see more RNA Pol II localized there. Okay, so for this reason, we call it expressed subtelomeric information. But it still doesn't tell us what the mechanism is. So we took advantage of, again, the kind of awesome power of yeast, where people, sort of armies of people in Toronto have now made not just every single deletion, but every double deletion, even there are triples, and started to make networks of what genetic relationships exist. We zoom into SNT1, and there are many, but one that struck us was RAP1, so kind of a classic epigenetic regulator that's important for keeping these regions of the genome silent, as they shouldn't ordinarily be on. And we wondered if this might be part of how the prion is acting. So we went back to do more CHIPs. Ordinarily, these regions are not bound by SNT1, the core, HOS2, the deacetylase, but they're decorated with RAP1. I've shown this one chromosome here, but I could show you any of them. The telomere is in green here. By contrast, if we look in the cells harboring the element, we see that there's tons of SNT1 and HOS2 there, and RAP1 has been effectively evicted, although it's maintained in the telomere. And so for this reason, we conclude that we wondered whether basically this coincidence might lead us to mechanism. So RAP1 is essential, but if we use an anchor-away technology to deplete it conditionally, we see basically the same behavior emerging. So increase in this RNA-seq reads in the subtelomeric region, as well as a phenocopy of the things that I showed you. But like all of this, the question is, what is it good for, right? It seems very complicated. It's interesting epigenetics. But what's fascinating is that as people have sequenced more and more fungi, and this is true actually in all organisms, that these regions that are repressed accumulate mutations much more than the rest of the genome. And that's probably for a couple of reasons, right? They're not expressed typically, so there may be less selection. And also, we know that there are all kinds of complicated sequences in those regions that lead to actually the production of more mutations during the replication. Okay? And so here's just an example from looking across 1,000 different natural sequencing of 1,000 different isolates of this particular organism. And what you find is that the frequency of polymorphism is going up quite a bit, and it coincides actually perfectly with the domain boundaries for these particular elements. And indeed, what we find is that across many different stressors, that the activation of those regions gives rise to pretty large changes in the phenotype. One of maybe the most exciting is the resistance to antifungal drugs that are used in the clinic. And some of you may be aware that indeed this is a big problem, that fungal pathogens are prodigious evolvers of this trait. And indeed, we wind up seeing this in isolates from patients who have resistant infections. And this turns out to be probably a property not just of fungi, but more broadly. So the other sort of major class of protein that we found were those that are involved in RNA binding and processing, and that won't surprise anyone in this room. And indeed, it's fortuitous that I speak because really one of the strongest hits was smog, which also exists in yeast, right, for reasons that were frankly unknown. And it has a lot of disorder and a tiny RNA binding domain. And that's true across all organisms. So it binds to SRE elements in the 3' UTR, recruits deadenylase machinery, and this winds up destabilizing the message, okay? And I don't have to evangelize you to its importance. Most biochemistry has been done on this very tiny ordered part of the protein. It's hard to purify the whole thing, right? But Anupam, who was doing this, thankfully was really an extraordinary biochemist and was able to get it to work. We wanted to try to figure out how it would bind to the transcriptome, but the problem is that the cross-linking approaches that we tried initially wound up basically stabilizing all kinds of higher-order conformers that we knew weren't present when we would do ordinarily blotting. So we did something a little bit more complicated, but that gave us much more control. We teamed up with Will Greenleaf to basically repurpose a sequencer to be able to look at binding to RNA. This looks kind of complicated, but basically the idea is you put a block on your library, and then you're able to basically transcribe it, and it's tethered above the sequence of DNA. You know the sequence of the RNA because you sequence the DNA beneath it. And this gives us basically kind of this idealized, synthetically transcribed genome where everything, whether it's normally transcribed as an ORF or not, is there in about 40x coverage. And we can flow on protein of known stoichiometry that we had complete control over and get 10 million measurements of on and off in an hour or two. Okay. There's a lot I could show you, but basically the punchline is that the disordered domain helps the protein bind much better. Okay? And that is kind of unlocking a new regulon, if you will. There are many targets that weren't ordinarily part of it. There's also an interesting thing happening with cooperativity. So if you look at just the RNA binding domain alone, you don't see, it's basically always have a coefficient of 1, but we see a lot of cooperative binding events for the full-length protein. And this is what's fascinating. They turn out to be in these giant clusters. So initially, we were filtering away large signals because we're worried they would be dirt, basically. And it turns out that you can be a little bit more permissive and get rid of real dirt, or what we would call like a false signal. And these large ones turn out to be the clusters that have this cooperative binding. And indeed, we think are related to the capacity to self-assemble. So if we look just here in vitro, what we find is that the RNA binding domain, we tag it with a Alexafluor, it remains soluble. But the full-length protein will eventually form these condensates. And they are happening at physiological concentrations. They're round, but again, they're not really quite liquid. So they will eventually fuse, but it takes days for this to occur. And they have pretty poor recovery in FRAP experiments. Our best guess is that they're gels from what we've done thus far, but we need to do more about that. And they bind RNA. Actually, they bind to the degree that you can compare the KD. They bind better, and they have, in fact, better specificity for SREs. This is just using a model FTSE-labeled nanosubstrate. Okay. And in fact, they can even recruit the deadenylase machinery better. So there are disorder domains on those proteins that wind up allowing them to co-localize into the gel. And you can see in the end, this leads in a reporter construct to actually better degradation. You can do a pulse chase and convince yourself of the same thing. And again, as I mentioned, this gives you kind of a couple hundred new targets that weren't there before, and they're not random. So if you look at them, they turn out to form a network. So the proteins that they encode interact with one another, and they control two things that are super important for this organism. So one is metabolism, and that turns out to be conserved in studies in Drosophila as well. And then meiosis, right? So again, this fascinating thing that this, or the most fascinating thing that this organism does. So we mix them 50-50. Again, these have the same genome. These guys have the heritable condensate. These ones don't. And they wind up winning over time. And as a frame of reference, this is a stronger selection coefficient than you see for most genes in this organism that aren't essential. So just one condensate that's there or not there. The opposite we see in the context of meiosis, so they're a little bit slower at making this decision. And you can link it all back to specific messages. So in this case, SRE element in MUM2, which winds up encoding another type of epigenetic modification, so basically an M6A modification in RNAs. And it just has a poor site that isn't ordinarily recognized, right? But now it can be with the better version of the repressor. It's a little bit complicated, but basically in this organism and many others, whether you decide to commit to meiosis or instead keep proliferating mitotically is linked to your nutritional status. So basically, if times get rough, now you're going to differentiate into a dormant spore. But you don't want to do it if nutrient is coming back. That would be a terrible strategy because your competitors that didn't do it, right, will win out. Okay? And basically what the condensate is doing is making this decision point more stringent. Okay? And people like Stan Leibler have spent a lot of time trying to model this type of behavior in the absence of a mechanism. They've argued that some kind of, if you will, a memory of the environmental fluctuations that the population has faced for an intermediate timescale, something that's shorter than genetic change, would be advantageous. And it struck us that this is maybe a mechanism for such a thing. And again, mathematically, it's clearly beneficial if you repeatedly have starvation that's going to be followed by nutrient repletion. But that's happening over a longer timescale than your lifespan, so it's hard for you to remember that. But that's, of course, exactly what we do in the lab, right? All the time, we're adding more nutrient, and reproducibly, we're going to do it. And indeed, laboratory strains of yeast are notoriously poor at entering meiosis. Everyone who studies this uses one particular background that in fact has a mutation in SMOG that makes it hard to adopt this state. And what we found was that if we play a trick, so we take these cells and we transiently inhibit chaperone, so remember that was required to make the seeds, the replicons to keep going, you wind up becoming much, much better sporulators. And through the magic of yeast genetics, we could show that that was basically due to the VTS1 or the SMOG element. So we can stop production of that protein for a little while and restore it, and then we eliminate the trait in the same way that we did with the chaperone inhibition. So it's not just a weird thing that we see in the laboratory. We also found it in isolates of the organism from the microbiomes of insects, from palm trees, all kinds of different niches around the world. And I think it tells us that this is not simply an artifact of laboratory cultivation. And actually, interestingly, the sort of types of assemblies that formed in those different isolates of the organism were different in character, as we've seen here today. You can get different conformers formed from exactly the same protein. Okay, so these elements do lots of other cool things. We see commonly that they reinforce other types of epigenetic modification, and we think that can help you kind of stabilize what would ordinarily be a transient response that would require reinitiation. We see cases in which, for example, they control whether in meiosis double-stranded breaks are resolved as crossovers or not, and a really interesting example controlling cell size and lifespan. I've also been curious, given the enrichment that we see in chromatin binding factors, whether they play a role, in fact, in controlling diversification of the genome. And indeed, it looks like that's true. Alex, who's here at the meeting, is working on this. You should ask her about that. And about this point, people ask me, what about in humans? They think there's something kind of special about yeast. And again, of course, all of you are probably aware that the sequence features I just told you about that give rise to this behavior are only enriched, right, in us. But we wondered whether it would be worthwhile looking. People have tried to find these types of elements either from amino acid enrichment, so that's kind of was Simon's claim to fame in his postdoc, or we've tried by this transient overexpression. But each of these presupposes some feature, right? Either that you look like an element that could do this before, or that your transient overexpression gives some phenotype that we chose to measure. So we wondered if we could look for something a little more generic, and again, return to the idea that all of these assemblies are a little bit stabilized in partial proteolysis. The degree matters, or can vary a lot. But they're all more stable than in their monomeric or native form. And so basically, we spent a long time trying to set up an assay like this. It wasn't simple. But basically, if you get a degron that is not especially potent, right, and you pay a lot of attention to the linkers, and you basically bracket your gene of interest by a selectable marker, or you can use GFP as well, and this degron, we can do the same transient overexpression trick that I showed you. And basically the idea here is that in the naive form, this is directional proteolysis is going to chew up everything. But in the assembled state, basically you wind up being inactive on this substrate. And so you preserve this activity of the selectable marker. And the linker was so important because basically you need to make sure that you still are active in the assembled state. Okay. So then we did this in yeast at the beginning simply because, again, you can capitalize on the ability to go through many, many different cell divisions. Okay? And basically, even if you assume an infinite half-life of the original protein, it's all diluted. Controls look good. Here's the classic prion protein. The activity, basically the ability to assemble, comes in domains. If we take off that domain, we wind up having a protein that isn't stabilized. And you can get about 5 orders of magnitude of selection in the experiment. So we deploy it on the human orpheome. And you can see 2 types of behaviors, right? So one I'm going to call an aggregate, but to be honest, we see this for proteins that are supposed to form condensates too. I don't think you can tell from the sequencing data which is which. But in this case, these 2 here are kind of controls, and the blue is the signal. So we see that after this transient overexpression, the protein is stabilized and substantially more than the rest of the proteome on average, and then it winds up being removed. By contrast, for something that can assemble and do so heritably, it winds up getting enriched in the population. And we do lots of controls to make sure that this is not from a mutation, and it's not because we happen to make more of this protein than another one. So we deploy it. We see lots of different hits. You can talk to Shady about FOXP2. What's cool is that we see many of them that are heritable over lots of generations. And we can play this trick again. Rather than using chaperone, we take advantage of our control over this promoter. Here we have a low level of expression that's just maintained over time. But in this experiment, we instead repress expression for a couple of generations, and then we bring it back. And what we find is that that resistance goes away. Okay? So basically, we have a transient elimination of the new protein, the new nucleotides of the system, basically, right? And now we're able to eliminate the trait. Okay, they form dots. They have interesting behaviors. And in vitro, they wind up usually forming something that looks pretty round at the beginning, and then over different amounts of times, they become branched. Those structures are pretty inert in FRAP experiments, and they have the capacity to template. Okay, so I've told you about a lot of benefits of templating. Obviously, there are costs. We typically associate this with various forms of neurodegeneration. And indeed, if we look at disease alleles of the proteins that we found in the screen, we see that they very often affect the phase behaviors. And often we see a case in which it's gained where it wasn't there before, or it's elaborated in the context of the disease mutant. Very often, actually, these are dominant diseases, and it's hard to maybe distinguish from the data that exists in the literature whether that's about haploinsufficiency, but it's intriguing to speculate, and in fact, we know from co-expression studies that the mutant can actually recruit the wild-type protein into the assembly. And we often think about these types of behaviors in the context of neurodegeneration, but in fact, if we look at the enrichments, we see even more for cancer and various other types of diseases. It's worth noting as well that cancer fusions very often mix and match these self-assembling domains and can change the behavior a lot in these assays. And again, you see a lot of what looks like a potential mechanism for genetic dominance, and in fact, that can be regulated by chaperone. So. But I think the real question is about how is this occurring in the context of aging, right? It's kind of the greatest risk factor for all of the diseases that have been associated with this type of behavior. Shown here is a microfluidic device where we've basically made traps for these cells, and they are budding off a daughter over and over again, and we can measure their lifespan by basically how many divisions they've gone through. And what we've noticed is that some prions, so this one is made by a pseudouridine synthase, wind up having all kinds of benefits in youth, but in age, they give you a reduced lifespan. And so we wondered if we would take a treatment that would erase all of them, so a transient inhibition of HSP70 chaperone, a generalist chaperone, then we restore function and look many generations later, what happens to lifespan. And this is an experiment actually I did myself while on sabbatical. I was very proud of it. What we wind up finding is that there's a pretty pronounced, increase in lifespan, and something different about the curve that I'll mention in one moment. Okay? So what we noticed, Alon, who has been on sabbatical this last year, is that this curve is both longer lived and also steeper. And that turns out to be extremely rare. So if you take lifespan curves from every organism, whether it be us or yeast or worms, flies, your favorite, what they do is they have a spectacular data collapse. So if you scale the points by the median lifespan, the curves have exactly the same shape. You can push things left or right, but there's nothing different is happening. And it's actually very few that change the shape. And so Uri wanted to try to understand that. And he built kind of a simple model of aging. It's agnostic to kind of what the mechanism is, but it turns out to explain this feature as well as kind of another fascinating feature of aging, that our risk of death basically increases with age until we reach some age, and then it's the same the longer that you lived at that point. And you can't just have accumulation of damage to get there. You basically need to have something you could think of as like a village, right, that's growing over time linearly, and that village makes some damage, and then that damage can be removed, and eventually you get rid of the removal capacity. You could think of that as like the aging factor that people often talk about. Okay. And the prediction winds up fitting data from the NHS, for example, extremely well, basically all data out there. But the prediction is basically that the mutants that affect damage will change the shape. So those that basically are the garbage trucks in the system, if you get rid of them, you'll become short-lived, and also the curve will be shallower. If you get rid of garbage, right, or you increase the capacity of the trucks, you wind up becoming long-lived and steep, right? Much like the manipulation I just showed you, where we got rid of these kinds of templating condensates. Okay. So we took the data that has been from an army of undergraduates, actually, at the University of Washington. Matt Kaberlein and Brian Kennedy had people dissecting these and trying to figure out the lifespan of every deletion mutant, many doubles, all kinds of things. And the question was, which ones wind up shifting it? And basically, there are exceedingly few. So the first thing that surprised us was that they're very bespoke. So if you look at the genes and protein quality control that do this, it's not HSF1 or these classic regulators of the whole response, or HSP90 or HSP70, classic chaperones. Instead, they're very specialized chaperones, the equivalent of DNA JB6, if you know it, that are involved in asymmetric retention of aggregates in mothers rather than being transmitted to daughters. Okay? And basically, they converge on a couple of things. So RNA binding proteins, in particular those that are involved in R loops, managing condensates, and then again, this asymmetric partitioning of aggregates. And of course, there are many other things, right, that change lifespan, but this is just what winds up converging in these shifts in the curve shape. And they have relationships to one another, right? So you'll see, for example, in a TOR mutant, that the shape can be shifted by a mutant in a DNA repair pathway that you might not have thought about. Okay. Again, is it yeast? Maybe not. It's hard to do these experiments in humans. But of course, we know that in the context of age-related neurodegeneration, protein aggregation is at least strongly associated with the disease. What about in normal aging, right? And in fact, we've often wondered whether this kind of self-templating might explain some of the spatiotemporal spread of pathology that has almost a quasi-developmental property, but it shouldn't, given the time in life when it's occurring after people have had their offspring. Okay, but of course, normal aging occurs over decades in us, and it's hard to study with an existing vertebrate model. Mice and zebrafish both take a very long time, right? And if you're a student who's impatient like me, you're not going to be very happy with your capacity to iterate. So my colleague Anne Brunet has pioneered a model that actually ecologists had known about for several decades. It's the shortest-lived vertebrate that can be bred in captivity. It's called the African turquoise killifish, but there are actually many species of killifish. This one lives in ephemeral ponds in Mozambique, and actually other places in East Africa as well. And in the context of several months, it goes through the same sort of transitions that you and I do, right? It matures, it can remember simple things, then it loses those memories. It also gets cataracts, cancer, and we see neuronal death as well in the model. And sort of the periods of lifespan in which those arise matches the fraction of our lifespan in which those traits arise as well. Yeah. And so we wanted to try to develop this as a platform to understand aggregation in the context of aging and potentially interactions among tissues in the process. Yeah. And we were helped here by Yuan, a chemical engineer in the lab who spent an extraordinary amount of time getting this right. She took those red cells and those white cells from the beginning, and she wanted the mass spec at the end to make it clear that the single protein that's in the aggregated form in the white cells and not in the red cells would be the hit from the mass spectrometry, even if you had no knowledge before what it was. Okay, so it's just a series of very particular differential centrifugations to get there. And what was fascinating is totally independently, she found proteins that assemble in each of these tissues, or I should say aggregate in these tissues. But they turn out to have extraordinary overlap with what we found in the Orpheum screen. Okay? And in fact, many of them that we have studied have the capacity to template in a prion-like way in the old tissue. This very interesting relationship between the biophysical parameters that are associated with aggregation within any one tissue and the demise of the protein quality control network that's specific to the tissue. For example, in the brain, we see loss of this DNA JB6 chaperone that I told you about, right? That acts on polyglutamine and polyasparagine proteins, and that winds up being one of the strongest enrichments that you see in the brain, but not in other tissues in which DNA JB6 activity appears to be intact throughout lifespan. Okay. Again, we can correlate these. There's a very interesting relationship to charge patterning and interspersal with hydrophobics that arises that appears to be really selective for the aging-dependent aggregates as opposed to those that aggregate throughout lifespan. I'll just show you briefly one example, RNA helicase called DDX5. We keep going through them. I guess we heard about 6 before, maybe 4 we'll hear about eventually. So this is a fascinating protein that, like I think the other 38 members of the dead box helicase family, has a known helicase domain and then often an RGD domain and a prion domain stuck onto it. It's important for the splicing of tau, so in loss-of-function mutants, you wind up getting a version of tau that has 4 repeats instead of 3. That we know is associated with being more aggregation prone. It's also really important for R-loop resolution and has many other phenotypes too. Okay. And again, it forms in vitro, viscous droplets that we can map. They wind up in phase portraits having transitions that correlate relatively well with the in vivo concentration. And it depends on the disorder domain, again, not a surprise. But it winds up also activating function. So it depends on the substrate you use, but basically from a calculation of how much extra this gives you, it's important to be able to catalyze the unwinding of substrates such as the tau RNA. But again, these will wind up maturing into a more disorganized aggregate that is inactive, and actually that inactivation is infectious. So if you do that seeding experiment, you will poison the entire pool. And again, potentially providing a mechanism for spread here. Okay. We see that this winds up happening in the context of old mice. This is in the brain here, and you see the formation of these cytoplasmic aggregates. What they're there for, we don't know, but one speculation is it may be for binding cytoplasmic R loops in this context. We also see this in samples from humans as well as exaggerated in AD patients. Okay. It's not just neurodegeneration. We see actually quite a wide variety of proteins linked to degenerative diseases wind up aggregating in this old animal. And in fact, often the Mendelian disease alleles associated with these conditions wind up in the context of the data I showed you earlier, giving rise to enhanced aggregation. So we think aging might be an interesting way in which these proteins that are perhaps predisposed to maturing from a condensate into an aggregate wind up getting accelerated. Okay, and then I just close our time together by talking about some exciting work from Shady, who will, I think, present a poster on it tomorrow, so you should go ask him about it. But non-vertebrates, right, have, so in plants, in bacteria, in fungi, they have this magic protein called HSP104. It's a triple ATPase that can reanimate an aggregate. And in fact, in rat brains where you look at synuclein aggregates, if you will deliver HSP104, you can rescue the pathology, you can rescue motor phenotypes, it's great. So it's really a shame that we don't have this protein. And I think it made us wonder whether aggregation is really just a one-way street in us, right? Or do we have ways to reverse it that just need to be unlocked? Okay. So Shady focused on polyQ, which is kind of a fascinating sequence linked to Huntington's disease, but actually many others. We know that having long tracks of polyQ is bad. And again, there are many diseases associated with this. If you look at across the proteome at consecutive amino acid length of each of the amino acids, you find that there are about 40 proteins that have, or the maximum rather is about 40 Qs. But there aren't very many proteins like that. 3 of them are shown here, so MAML2, TBP, and FOXP2 is the most exaggerated one. And each of these other proteins is associated with a polyQ-opathy, but FOXP2 isn't. This is the so-called speech gene that's received a lot of press, very controversial. But basically, there are it's an extremely conserved, intrinsically disordered region relative to others that we've heard about at this meeting. And there are 2 mutations that we have that chimps don't, that actually are in this region. And many people have tried to understand basically whether those are in part responsible for our capacity to speak. It was found because of a family that has dyspraxia, but no obvious other challenge in terms of cognition. And in fact, if you take the human allele and put it into mice, they will vocalize more. In any case, fascinating protein. How could it have so many polyQs and they're conserved? The family mutation, and this one as well, basically abrogates binding to DNA. So that's not surprising, kind of a loss of function, but they form aggregates when they're not stuck on DNA. So one way in which we think that you're able to maintain this is just by, in fact, using all that charge to help you out. And in fact, if you take these aggregating mutants and stick synthetic DNA binding domains onto them, you can rescue solubility. And this turns out to be true not for all transcription factors, but for many of them that we look at, that if you make mutants that don't bind DNA, they will aggregate, and then you can rescue that by making them bind to DNA again. But this raised another question, which is what happens in mitosis when we're not bound to DNA? And indeed, we saw a surprise. So normally you don't see this often in culture, but what we found was that cells that we could catch in mitosis have soluble FOXP2. And in fact, that we could see this for all kinds of different alleles, whether that be the one found in the family or others that we generated that would form aggregates by having, for example, this phosphosite mutation. Doesn't depend on DNA binding, just the N-terminus of the protein that has only the disordered domain is also capable of being dissolved. And we kind of wonder if it's related in some way to the fact that these diseases arise in post-mitotic cells. Again, we see this for Huntington itself as well, and are engaged in some IPMS and screens to try to identify the factors that are involved. We have some promising candidates. If you want to hear more about this, you should talk to Shady tomorrow. Okay, and with that, I conclude. I think this type of information transfer, I shouldn't just say beyond the chromosome because plenty of it is happening on the chromosome too, is more common than I would have ever predicted. And a lot of our prior assumptions about it are just wrong. It's been amazing to see it. This probably provides an economy for the proteome. You can get more than one activity out of the same polypeptide. Alternative splicing is basically mixing and matching these domains more than any other. But it raises profound questions about what the native fold is, right? So that decision between whether to proliferate mitotically or meiotically in yeast has been studied hundreds of times, right? And people missed it, right? And in fact, what we think of as the native fold of SMOG in this organism is the templating fold. So it tells us that kind of the history of your lineage matters for how a protein is made. And again, I think it gives us a kind of new paradigm for how kind of a layer of gene regulation, maybe stabilizing other types of epigenetics, and much, much more in terms of, again, giving you new regulatory opportunities. And then finally, I just comment that these sequences, by virtue of the sequences that encode them, and the DNA sequences that encode them, are evolving more rapidly than the rest of the proteome. And that can lead to kind of interesting changes, right, in the phase behavior over evolutionary time that perhaps could be useful in terms of innovation. But also, clearly in the context of aging, things can go bad. And once they do, it can be really a problem. And with that, I thank you for your attention. The folks in the lab who did the work, as well as collaborators, I tried to highlight them along the way, and then the kind folks who funded us. Be happy to take any questions. Simon. Oh, no, students first. No, it's okay. That's sorry. No, please, yes, students first. Yeah. I just don't know your name, so that's all.
Hi. Stand up. Okay. I'm Chris Johnstone from MIT. This is very interesting, and I was wondering, for these newly recognized regulated prions in yeast, do you ever see that phenotype reversing, and by what mechanisms do those reverse?
It's a great question. So, yeah, for us to use, can you lose it, right? And yes, so the spontaneous rate of loss is roughly equivalent to the rate of gain. So usually 1 in 100,000, 1 in 10,000, depending on the particular element. But we have also seen examples of induced loss. So for example, one that I didn't talk about today, if you desiccate the cells, it will selectively be eliminated. If you think about kind of what features you need to keep the cycle of replication going, right? Losing the protein expression for a cell cycle causes you to get rid of it. You can't make any more of the assembly. And then as well, we know that phosphorylation within the disordered regions will basically reorganize them in ways that make the nucleation sequence either present or concealed. And so you can also make phosphovariants that are unable to be propagated. So, but it's an interesting area for future discovery. We know that they have to be lost, right? Because most of them, unlike smog, are not present in all cells.
I really liked your stuff on ESI1 because I found something similar in like a telomerase knockout strain. Cool. I don't know if you looked at that pathway, like if it affected aging in like a TLC1 knockout strain.
We haven't looked at the interaction with telomerase mutants per se, although it's on the docket. We thought it would be short-lived, but in fact the opposite is true. It's one of the few prions that we looked at that extends lifespan. And again, the mechanism, still not totally clear. We've made a bunch of doubles trying to understand all of the contributors, but yeah, it's a super cool question.
Yeah, Dan, so for SMOG and DDX5, you see these assemblies, and they're both RNA binding proteins. So when you have RNA present, do they still form? I mean, from the beginning now, not adding it after the assembly, but before.
Yeah, so for DDX5, but basically total RNA doesn't do anything. We have not made synthetic R loops that I think would be interesting to look at. For SMOG, I think we've done total. It didn't change anything, but again, I think we should try the SRE elements. So if we take from that array experiment I showed you on the chip with all of the red dots, some messages from there look like they're able to help seeding happen initially, but we haven't explored it. There's something kind of complicated about the spacing of the loops, basically, that appears to matter that we don't fully understand. But I think we don't have evidence for enormous effects, but I think in the case of SMOG, it may be that particular messages have SREs that are spaced appropriately that kind of help to push things along.
I mean, the reason I'm also asking is, could there be a mismatch between the RNA concentration, also the target RNAs, and the proteins, and could that be relevant for aging, for example? Exactly. I mean, with age, you have, we know that transcription was haywire, and there's changes in RNA targets. Could that be a mechanism for how it all gets going?
Certainly, I mean, yeah. Obviously, changing the concentration of RNA could matter a great deal, right, for many of these. In general, they tend to be better at binding, so I guess our naive assumption would be that you are buffered against it, but we should think more about it. It's a good question.
Hi. So in the end, you mentioned the evolution of those IDR, and that's very interesting because we know those sequences usually evolve very rapidly. And so I wonder, have you looked at some close species to the Kilifish, which doesn't age that fast, and compare those IDR versus the one you found. Anything you can comment on?
That's a fabulous, I didn't plan to, but that's, so, yeah, the challenge of working, how to begin this? You know, it's often people are told to like find a prodigy, right, when you want to study something, not just necessarily a model for humans, but you know, something that does something well. And I think, you know, we like yeast because it's really good at cell division, right? And if we want to study transmission of information across cell divisions, that's a great spot. You know, here, this is obviously an aging prodigy. And as I mentioned, there are many other killifish species that have different lifespans, in fact, you know, ranging up to about 2 years. You know, the challenge of working in an organism that hasn't been studied well, like mice, is that the tools are not great, right? And you know, only very recently, within months, right, have the sequences of those organisms become available. But indeed, we are actively looking to try to understand basically if there are genes that we found or proteins that we found that show variation in a way that makes sense in light of the lifespan. It's likely that it's going to be an emergent property from many of them, so you know, it may not immediately come from it, but it's a great idea that we are engaged in.
Hi, I'm from Dan's lab, but I did not get planted for this question. I wasn't aware of the data that you mentioned where the exogenous expression of HSP104 can rescue some of these neurodegenerative phenotypes. And so I'm curious from like an evolutionary biology perspective, what kinds of selective pressure would lead to the loss of HSP104 if there's convergent evolutionary loss of this disaggregase?
Yeah, this is another good question. I think it's probably just like a low effective population size and drift. But yeah, I think that's probably what it boils down to, rather than selection for not having it. Yeah, the mice, or the rats, sorry, in that experiment, there's nothing obviously wrong with them. I don't know that people have looked in great detail. It's another good question.
Hi, Katie Galloway, MIT. I found your talk really interesting, especially the aging curves, where you see there's only a few conditions where you change the shape. I was curious, when you say you have to have this steep drop-off, is that because you've constrained the number of generations, or is there some other feature that's actually specifying that steep drop-off?
Yeah, so in the model, there's a threshold for the amount of damage that you accumulate before you wind up hitting death. And so basically, yeah, when you are long-lived, you're kind of increasing the the removal, right, of the damage, but you're going to wind up hitting it very steeply because there's sort of less noise that you have to get there. You're going to push up to the threshold is what winds up happening. And again, empirically, it's fascinating how much this winds up working.
So a simple question. You use 3 terms, prion, condensate, and aggregate.
Yes.
And can you define why you use different terms for different phenomena?
Yes, good. So it's an excellent question, one that I struggle with a lot. So I use condensate pretty generally. I use aggregate to describe something that I think of as pretty solid. And prion is a tough one, right? It stands, Prusiner was very wise to define in his 1982 paper exactly what it meant. It was a proteinaceous infectious particle. It doesn't suppose anything about it being amyloid at all. And the challenge is what you mean by infectious. So I think sort of high church prion people would argue that only PRP qualifies, right? It's just shed from animals in the urine of sheep, for example. It will get stuck on a weed in Sardinia, and an animal can come along months later and have it. None of these are at that level, but certainly in fungi, right, in sex, the trait is transmitted, the aggregates are transmitted. So I think there's something that we often add on to this, sort of prion-like, for a set of molecules that have the type of self-templating, but maybe by virtue of either a barrier that confines them to the cell in which they arise, or just the protein only being expressed within a tissue or particular cell type, the capacity to transmit is limited relative to the example for PRP.
But your aggregates and your condensate transmit information.
That's right, and those I would call them prions. That's right,
Okay.
as well. I wouldn't, yeah, I would. I think 6 months into my job, I was giving a talk at Cold Spring Harbor, and Sam Prusner stood up and yelled in the middle, like, why aren't you calling them prions? So I used to have other names for them, but I changed my tune.
Other questions? If not, let's thank Daniel for a fascinating talk.
There's one.
Oh, please. Go ahead.
But you don't have to applaud twice. That's okay.
Oh, you deserve it.
So I'm a PhD student from Epigenetics in Switzerland. So I have a question. For me, it's hard to understand why DNA binding to RNA binding proteins can restrict solubility, and also mentioned that DNA binding to this kind of proteins can also enhance binding affinity. So normally, I think the DNA only binds to the RNA binding domain, right? So what do you think is cooperativity? And do you think the binding of DNA will change the structure of the protein? Is it still disordered or not?
Great. That's very good. Which is the first? I take the second one first. Yeah, I think intrinsically disordered is a wastebasket diagnosis. All of the evidence we have about, much of the evidence we have about these suggests that they are actually quite ordered in the cases in which we study them. So I think of it maybe more in this instance as being a marker of the lability of the structure. For the, what's giving rise to the cooperativity, we don't know. One possibility is that actually there's sort of some preformed complex. We have some evidence for this from analytical ultracentrifugation and size exclusion. It doesn't look like a single peak. It's some sort of broader ensemble. And we've done a little bit of modeling to suggest that that might wind up giving you more cooperativity, but that part is still pretty nascent. You notice it is in preparation. For the DNA binding, right, that's one question about whether it is just the polyanion, right? It's not something special about the sequence that FOXP2 binds, because you can get the behavior from adding on, right, these other DNA binding motifs that have different sequence specificity.
Okay, thanks.
Okay. Thank you.